Okay, finally we have reached photosynthesis part 3, the Calvin cycle. Here we are going to actually make some sugar. So let's kind of refresh where we are. We're picking up after the light-dependent reactions. And here is what we have. We have our ATP. This is our sort of captured chemical energy for the cell. NADPH, carrying with it its electrons or reducing power. And oxygen gas has already been released. We're not dealing with that anymore. It's been released through the leaf's stomata out into the atmosphere, where it can be used by creatures like us. And we have successfully trapped the sunlight energy and put it into a chemical form, but it is not yet in the form that we need. So the cells need glucose. We don't have glucose yet. To orient ourselves physically, we were in the thylakoid membrane and a little bit in that lumen with the concentration gradient. We now move out into the stroma. Remember, this is the fluid that surrounds those grana of thylakoids within the chloroplast. Don't be afraid of the diagram you are now looking upon with horror. This is a lot more detail than you need to know. I did want to show it to you just in case you're curious, but also because there's a decent chance that a diagram like this will be presented to you on an exam question, and I don't want you to be like, oh no, I just want you to be like, oh yeah, I know what this is, and I know I don't have to understand every little tiny piece of this. Before I go further, before I forget, I just wanted to mention I'm going to be using Calvin's cycle. These reactions are also called the dark reactions sometimes, and also the light independent reactions. And they're called the light independent reactions because they don't require light. And in fact, even though sometimes they're called the dark reactions, they often will slow down or stop in the dark because, of course, they're dependent on products of the light reactions. And the light reactions require light. And if there's no light, then they can't, those light reactions can't make the ingredients needed for the Calvin cycle. Calvin, of course, is just the dude who discovered this. So anyway, all right. So let's look at the stuff that you do need to know about the Calvin cycle. Of course, you absolutely need to know that we are making glucose during this phase of photosynthesis. But in terms of the parts of the cycle, you need to know the main stages or phases of the cycle. So we will go through each of these and I will give you the details that you do need to know and not a lot more, okay? So those stages are carbon fixation, reduction, and then regeneration of ribulose bisphosphate or RuBP. Okay, so carbon fixation we're doing first. First, you need to understand what that word means. You'll hear it sometimes in relation to carbon fixation like here, or sometimes nitrogen fixation because certain organisms or certain bacteria that can fix nitrogen. And all that means is they can take a biologically unusable form of something and turn it into a biologically usable form for themselves. And in this case, it refers to taking carbon dioxide, which is not directly usable by the cell, and turning it into something that they can use, okay? For this phase, what's going to happen is an enzyme called Rubisco, that's an abbreviation that stands for ribulose bisphosphate carboxylase oxygenase, I think. But anyway, it's called Rubisco because that name is a mouthful. This enzyme will do the carbon fixation, and it does this by adding carbon dioxide to a pre-existing 5-carbon molecule called ribulose bisphosphate, or RuBP. So I'm going to show you a picture because I know that will help in a second. But essentially, it's adding this carbon dioxide molecule, and there's a lot of mixing around, moving around of atoms and stuff that we don't need to worry about. You need to know that that carbon dioxide is added to RuBP in creating this sort of transition molecule that has 6 carbons. That molecule is unstable and will quickly break down into 2,3 carbon molecules called 3-PGA, but you should know that this has now changed this unusable carbon dioxide into a form, an organic form that the cell can work with. Also file away, please, that in addition to bringing in the carbon, which is what we focus on a bunch in this cycle, it's also bringing in the oxygen from carbon dioxide. So while the oxygen in the atmosphere comes from the splitting of water in the light reactions that we already talked about, the oxygen in glucose comes from carbon dioxide during this process. That is a tricky thing that I do see asked about. So you should know where the oxygen comes from for glucose and for the atmosphere. Okay, let's move on. Here's a picture of what I was just telling you about. This is, and again, you don't need to know molecular structures, but just to kind of give you a little idea. On the sort of top left here of that circled portion of the cycle, you see RuBP. You see the Rubisco enzyme here is acting. That's why that name is there. We're bringing in carbon dioxide from the atmosphere. Of course, that came from the leaf stomata. And it's going to be combined. Things are going to be, switched around a little bit until you ultimately end up with 2-3 carbon organic molecules that can be used for this next step. So we have successfully turned carbon into a biologically usable form, brought in that carbon and its oxygen, and turned it into 2-3 carbon molecules. That's stage 1. Next, we're going to do some reducing. Who has the power to do some reduction? That would be NADPH. We're going to use the energy from ATP and the reducing power of NADPH from the light reaction. So the whole shebang before was about creating a bunch of ATP and NADPH, and now we're going to use those ingredients. ATP and NADPH are going to reduce or give electrons to the 3-PGA. So remember, Leo goes GER, gain electrons, reduction, giving electrons is reducing. So, or gaining electrons is reducing. So this PGA is going to be reduced with some little intermediary steps, etc. Ultimately, this is going to make a 3-carbon molecule called G3P. Some of this G3P will exit the cycle and be used to make sugar, and other G3P molecules will be used to regenerate the RuBP that we used in stage 1 so that the cycle can begin again. Let's look at this in picture form. So we already did the carbon fixation at the top. Now we're down here where it says reduction. And you can see we are using our ATP and we're using our NADPH to make G3P. Some of that G3P, you can see that arrow going out and down, is going to exit the cycle and be used to make glucose. And other G3P molecules are going to move into the regeneration phase and be used to regenerate the ribulose bisphosphate that we need to start the cycle again. I want to make one note. You may not have even noticed this, but if you've noticed that sometimes pictures of the Calvin cycle start with 3 carbon dioxides, sometimes 6 carbon dioxides, and sometimes 1, here is why. It takes 3, you get 1 carbon dioxide from each of these cycles for glucose. So it takes 6 turns of the Calvin cycle to make 1 glucose molecule. Remember, glucose is C6H12O6. C6 means there are 6 carbons. So that is why. If it is showing, sometimes it will show like this one, how many turns you need to make 1 G3P. That's 3. That's why this one's 3. Sometimes it'll say 6 carbon dioxides to show you how many of each of these molecules are involved in creating one whole glucose molecule, and occasionally you'll see it with just one carbon dioxide molecule and just showing sort of the path of that one molecule. But I don't want you to see the different numbers and be confused. You don't, for this particular process, need to know numbers. You just need to know the general sort of what's happening at each stage kind of stuff. But just in case, I didn't want that to confuse you. All right, so we have some of that G3P having exited the cycle and on to make glucose. Fantastic. And then we need to use other molecules of G3P to regenerate ribulose bisphosphate. And so it's basically all you need to really know is we're going to use ATP energy from the light reactions to do a little molecular rearrangement and regenerate that RuBP. And then the whole thing can start again. So just looking here again at the diagram, you can just see we are in the regeneration phase. We're using some ATP. Here's one. You can notice this diagram has 6 CO2 at the top. We're using some ATP to regenerate that RuBP, and then we can start again at stage 1. So that was, kind of a lot of information, not as bad as the light reactions. It is probably going to be something that you need to go over again in practice. It's just, they're just like a lot of weird names of things. And so the processes are kind of unusual. Give yourself that time now. Spend the time before we move on to the next process that has, similar esoteric weird things that you have to understand, so that you aren't trying to, learn too much at once. Okay, so go over this if needed. And before I leave you, I want to do a little quick check. So here is our overall chemical reaction for photosynthesis, because we've reached the end. And I want you to ask yourself if you can answer these things. So you're going to pause and see if you can answer. Where does the oxygen in the atmosphere come from? Okay, so where does it come from? It comes from the splitting of water. During what stage of photosynthesis? The light reactions. Okay, then number 2, the oxygen and glucose. Where does it come from? Yep, that comes from carbon dioxide, and that is from the Calvin cycle. Where does the carbon in glucose come from? Again, carbon dioxide in the Calvin cycle. And then this one is more just a connection between the two phases. We have used up a bunch of ATP and NADPH. That means that we have generated a bunch of ADP and NADP plus. Where does that go? Just think for a second. Yes, of course, it has to go back into the light reactions because those are needed so that we can create more ATP and NADPH and start the whole shebang again. Okay, come with questions and be ready to discuss. Excellent job.